The overall goal of this procedure is to quantify acute changes in neuronal surface proteins in ex vivo brain slice preparations. This is accomplished by first preparing viable acute brain slices. Next, the slices are treated with drugs of interest. Then, the cell surface proteins are labeled using membrane-impermeant biotinylation reagents. Finally, surface biotinylated proteins are isolated using streptavidin affinity chromatography. Ultimately, immunoblotting is used to quantify changes in protein surface expression following drug treatments.
The main advantage of this technique over existing methods, such as cell-based biotinylation, is that neuronal protein trafficking is examined in situ rather than in heterologous expression systems or primary neuronal cultures. Generally, individuals new to this method will struggle to prepare viable brain slices.
To prepare brain slices, begin by making fresh 1x artificial cerebral spinal fluid, or ACSF, and sucrose-supplemented ACSF, or SACSF, according to the text protocol. Chill the SACSF on ice. Use 95% oxygen with 5% carbon dioxide to saturate the buffers with oxygen by bubbling for 20 minutes on ice. After sacrificing P30 to 38 mice by cervical dislocation and decapitation and rapidly removing the brains, place them in pre-chilled oxygenated SACSF. Using a vibrating microtome, make 300 micron brain sections in the region of interest.